About to record. Right, so we'll start with the diagram of animal cell first, then we'll go to plant cell. Animal cell. Okay. So animal cell is usually round in shape, so you can just draw one big. Circle kind of a thing. Does not have to be exact circle, but okay, if I do this, everything will be erased. Right, so make something like sort of a, first thing what you'll do is you draw ovalish shape. Let's call it oval. Right, first draw that. Then we'll start drawing the organelles inside. Okay. Next we can draw nucleus, since that would be the biggest. Okay, if I draw nucleus here. Okay, and next to it, you have your endoplasmic reticulum. So endoplasmic reticulum, usually they make it like this. So if your first, what is this called? If your first, a pathway or your first lane is here, then the other one will be here like this, opposite they draw. No reason, I think just to make it neat, I guess, only for that. No particular reason for that, but yeah. So right, and then finally we can just yeah, you can make the same thing this side as well. I'm taking some amount of time because not able to draw with this. So usually in animal cell, the endoplasmic reticulum will take up a lot of space, whereas in plant cell, the vacuole takes up a lot of space. So plant cell is easier to draw always. Right, something like this. Okay. Okay, if you want, we can extend this one a little bit towards the end. Yeah, something like this. Just to show you that basically it will be touching both ends, or traveling almost to the edges of the cell. Okay. Now we have to add our nucleolus inside. I am just adding with different colors so that you all know. You can draw it with one, normal pencil. Not an issue at all. Okay, let me also keep it one color only. Leave it. Okay, and the nucleolus inside here. Right, done. Next we have to draw, okay, we can draw any of them. We can draw Golgi apparatus. So Golgi apparatus will be something like this. Okay, so basically these keep increasing in length. You can draw just 3, 4 and just draw some 2, 3 dots next to it like this. Right. Next we have, of course, we have to draw mitochondria. Mitochondria would be like this. Make sure you're making a little bit big enough oval because you have to draw the folds inside as well, right? So make 2 because mitochondria has 2 membranes. Right, and then we'll just make folds inside here. Something like this. Okay. Make 2, 3 of them. Make one here, make one here also maybe. Okay, maybe let's make one here.
This is mitochondria, right? The one you
Yes,
have
yes.
made.
Yes.
So we can make it anywhere in the
Yeah.
cytoplasm.
Yes, anywhere in cytoplasm. Anywhere. Golgi apparatus also anywhere. Just keep it little closer to the endoplasmic reticulum, but you can make it in the bottom half also. Anywhere. Only fixed location is the endoplasmic reticulum. It has to be next to the nucleus. Everything else can be anywhere. Okay, I'll label it also now only because that seems like a better idea. Right? Oh, I forgot. I made all this, but I forgot to put the rough endoplasmic reticulum part. Right. Okay. Or Golgi body or Golgi complex. Right. Then this is our nucleus. Actually, nucleus also you can make one more membrane inside because nucleus also has nuclear membrane. If possible, you can. I'll let it. It's good enough. Okay. Just let me check.
There's only these parts we had to draw.
Yeah, and of course you'll draw multiple mitochondria, but there's nothing more. One more is lysosome, which we have to make. Lysosome.
Miss, how many mitochondria should we make?
2, 3 is sufficient.
2 is sufficient.
Yeah, 2 is sufficient. You made a tiny cell, 2 is sufficient. If your cell looks empty, make one more.
Okay.
Right. Okay. So this will become your ribosome. Correct. This becomes rough. Okay, I'm erasing this. This is no point to write steps. Yeah, rough endoplasmic reticulum, correct? And हाँ, smooth भी चाहिए ना, smooth available है, smooth endoplasmic. For labelings, you can follow any convention. Either label all of them on right hand side. If you feel it is getting cluttered or you're not left with much space to label, then go to the left and spread it out. So that all doesn't really matter. Just make sure that your diagram is neat. That has to be, that is of utmost importance. You can put this also labeling Nucleolus. And as far as possible, try to avoid overlapping things. Like don't make an arrow from like this to the nucleus. Okay, avoid this. You know what you can do is you can bring an arrow from the bottom so that it's not overlapping anything. It's a neat way to do. Right, and of course you can make arrows also. If you don't make also chalega. Because sometimes they say draw the arrow in the direction of the organelle. Sometimes they say draw it where you have written the names. So if you're not making the arrows also it's fine.
How many, like, can we make 2 or 3 mitochondria or?
Yes,
like,
yes. Two or three, yes. Don't make only one, make two or three. Chalega. If you made a very large cell, you can make four also. That's also fine. Just make sure your cell is, nicely distributed. Everything should not be on one side, or it should not be empty on one side. Something like that, don't do. Other than that, anywhere you're making anything does not really matter.
Is this fine?
1 second, I can't see. Yeah, looks fine. Correct. Okay. Yeah, make sure to make some ribosomes as well floating around. And we've forgotten we've forgotten lysosome, and we've forgotten vacuole also. Two or more are there, right? So make one lysosome somewhere and one vacuole wherever you have space. So I'm making only two mitochondria because if I make one more, then somewhere it will get. Actually, I can. Okay, I'll make one more here. Actually. Okay, more I made here. Okay. And I have to make one vacuole. I'll make vacuole here. Spread the ribosomes a little bit around. Right. Okay. And now I'm left with lysosome. Lysosome, I can make it here. There is empty space I can see here. So I'll make it here. Wherever you see empty space, just make one small lysosome. Right, lysosome and vacuole will look the same. There's not much you can do
Miss,
about
what are
it.
the dots for those dots?
Ribosomes there. I
Achha.
will make it again here. Ribosomes. This is important because this shows that you know that it is not only attached to the rough endoplasmic reticulum, but also found freely in the cell. So when you draw this, you'll automatically get bonus point. That okay, you know. Right. What am I missing? Anything? Am I? No. Okay, they've made lysosome like this. Why I don't know, but okay, chalo, can also make it to distinguish from the vacuole. Okay, yeah. So this becomes my lysosome, correct? This I will label as vacuole. This I will label mitochondria. Just any one of them. You don't have to draw arrows for all three. Just pick up any one which is easiest for you to label and put mitochondria. Correct? We've also forgotten cell membranes. Cell membrane, I'll put it here. Right? Anything else I missed?
Miss, do lysosomes have a particular place?
No, no. Only nucleus will be, somewhere around the center of the cell, and endoplasmic reticulum has to be next to the nucleus. Everything else can be anywhere, not really fixed. It can be anywhere.
Okay.
Okay, I made, yeah, you can label cytoplasm also. Right. So just anywhere where you see free space, here there is more space. So I'll make it here. Call this as cytoplasm. Correct. Anything else am I missing? I think fully not. Let me just check. Ribosomes.
Miss, it's necessary to label on only one side, right?
Okay. That actually depends, unless they're specifically told you one side, you can keep it both sides actually. Have they told you in school label one side only?
Yes.
Then label one side only. Label everything on either right side or left side. Follow that.
But Miss, I labeled everything on right side. I don't have more space.
Then put an arrow like this now. Make it like this, right angle type, and put it.
Miss, that's only what I'm doing.
Haan.
So वैसे if I make it on left side, is it fine?
Yeah, it's fine. No worries. Okay. Did I miss anything? Did I nucleus covered? Nucleolus covered. Endoplasmic reticulum, mitochondria. Acha, they have put mitochondrion next to it. Okay. Put that also. Golgi body, lysosome. Yeah, somewhere, I noticed one more thing. Somewhere in if you see the internet or even your chapter notes, they have drawn the nucleus like this. Okay, the nucleus is here. Then the endoplasmic reticulum is like this. The channels are not attached to the nucleus. If you make it like this also is okay. So everywhere you will see a new variation of the diagram. So all the variations are fine. Just make sure you're, labeling whatever you're drawing correctly. As long as the major parts are matching, it does not matter much. Chondria.
Miss, that's the nucleoplasm, right?
This
With
thing?
the nucleus.
Yeah. Yes, it would be the nuclear membrane. Nuclear membrane. This would be the nucleoplasm. In between the nucleus and nucleolus. Because cytoplasm, there's nucleoplasm inside the nucleus. But yeah, not necessary for labeling all that. Nuclear membrane also is not required.
Is there anything inside the nucleoplasm?
You mean here? Here,
Yeah,
this would be a
like,
nucleoplasm.
example, in cytoplasm, there's everything. So, is there anything in nucleoplasm?
I'm not really sure because the chromosomes are there inside the nucleolus. I will check and tell you if there's anything in the nucleoplasm. I'm not sure if there is anything.
Miss, the outer one is nuclear membrane, right?
Yeah, this part. This part is the nuclear membrane.
हाँ.
ठीक है. Okay. Okay. Tell me once you all are done, then we'll go on to plants.
Done.
Done. Okay. Done? Okay. All of you are nodding your heads. Okay. ठीक है. I'll be a little take a shorter route here. I will leave the inside part as it is. I'll just change the outer membrane. Right. Sometimes for mitochondria also, na, they have written mitochondrion. That is also correct. So mitochondria usually is plural. Okay. Mitochondria. Instead of this, you will find somewhere labeled as mitochondrion. This is referring to singular actually. That's all. But even if you have written mitochondria, chalega. Right. This is if you come across some diagram and you see these words, you might wonder which to use. So for that, I'm telling you.
Miss, mitochondria चलेगा ना?
हाँ, chalega. Yes. Sometimes they'll ask you to draw the cell and label some particular things only. Okay. So if they ask you some question like that, make sure that you are labeling whatever they have asked you to label. Yeah, I need to remove this. I had to remove that. Why I remove this? That's it. Okay, here I'll make a different color. Plant cell is usually rectangular or more on the sharper side. So you can make rectangle or you can make hexagon like this. This is difficult to make hexagon. You end up spending more time making it like right, and hexagon or pentagon, whatever. Something like this, or you can just keep it simple and make it a little bit square like this. So whichever, some diagrams you'll see with the cell wall like this, some you'll see like this. So whichever you find easier to draw, always stick to that because there's no compulsion that you have to make it look like this or like that. Yeah, I find the rectangle version easier, so I'll just draw that. Again, rectangle one also has variations where one they will make, they put like this. When I was drawing the diagrams, I made like this, no, for your. So this also is optional. You can ignore this or you can add these. Up to you. Okay, so I have to make it rectangular. I will make it like this. Oh, actually, this will not work for me. Okay, I erase. I have to erase everything. Sad. Okay, and another we'll make another one inside, for which would be our cell membrane, right? Cell wall, again they draw it sometimes like this. So this would become the cell wall. Just thick. Okay, so this becomes, okay, this time I'll also label it this side only. It's easier for you. This cell wall, correct? Okay, now inside we have to make one more, which will be our cell membrane. You can leave some space and you can make a membrane like this. Right. So then this becomes the cell membrane. Okay, let's label it. Correct. Now you next thing you will make would be the vacuole, which would be the biggest part. So here once you make the vacuole, then you can make the remaining things next to it. So vacuole will be the largest part, usually like this. Again, it can be any shape. There's no fixed shape. Just don't try to make it a perfect square. Okay, keep it rounded edges. Whatever shape you're making, just make it rounded. Okay. Yeah. Here also don't try to make perfect squares. Whatever you're making, no, make it little rounded. Because there's no fixed, exact, precise geometry in biology or in nature. You'll get some kind of a square kind of shape, but it will not be a straight line anytime. So ensure all those things. Okay. Right, so this is your vacuole. Next important one. Correct? Okay, now I'll make things around the vacuole. If there is more space here, I'll make the nucleus here. Okay, this time let us make different version of the nucleus. Right, and you have nuclear membrane like this. Nuclear membrane is usually like this with pores in between. And let's say you have nucleolus. Okay. Now here I will draw the endoplasmic reticulum not attached. So that you have, you get an overview of both types, how it looks. You can follow anyone. Okay, let it be. I'll draw attached. That is better. Okay, this side I'll make little smaller one because we have to accommodate lot many things here, no? So we'll keep smaller one. Okay, next thing would be to make ribosomes. Correct. Okay. Now you make Golgi apparatus somewhere, wherever you have space. I have more space here, so I'm making it here. Wherever you have more space, make the Golgi apparatus there because that is usually the biggest one, right? Let me start small. Okay. Okay, I have to label these also, right? Label. Nucleolus. This is okay. I'm putting smooth endoplasmic reticulum. Nucleolus, right? Then we have uh, rough endoplasmic reticulum. Then we have yeah, I forgot nucleus. Put that. Nucleus, right? Okay, and I will make some chloroplast here. Now, important in plant cell is chloroplast, right? I will not be drawing lysosome. I will be drawing chloroplast and mitochondria. So wherever you have space, just draw 2, 3. Like I can fit one here. Okay. Both are double membrane. So make sure you're putting 2 membranes like this. Right, let's make mitochondria here. Yeah. Okay. Then I can make one chloroplast here. And one more here. Make this one bigger so that you can see what to draw inside. Right, so inside we can make stacks. How to make those stacks? You can make. Okay, let's just put it like that. Okay, we make like this. Okay, I'm drawing the enlarged version here in case you're not able to see that. Made this, I made another covering. Okay, and inside I'm making the stacks connected with each other like this. Okay, in the chapter notes and in the on the internet, they've just drawn something like this. But avoid doing that because you can easily draw this note. So to avoid doing that, make it like this one. Right. So if it is smaller, make smaller stacks. Okay, done. So stack is enough. And draw one more here. I'll draw one mitochondria here. What else are we missing out on? Ribosomes. Anything else? Something else. Yeah. Ribosomes. Okay, so this is how your plant cell should look like. See, there is everything is filled. No, it's not. I have not put all the organelles to one side, or I have not left major empty space in between. It looks filled. This much is sufficient. You don't have to make sure to fill each and every gap or something like that. Just make sure overall it looks like a complete thing. Okay, let us label the remaining things as well. Okay. This would be Golgi Apparatus. Correct. This would be Chloroplast. Okay. This would be Ribosome. Okay. What else? Did I miss anything? I miss mitochondria. Correct. For mitochondria, I have no space left, so I'll label it here. Okay. Did I forget to label anything? Then let me know, because I might have skipped something. Cytoplasm.
Done, miss.
What about Ashmi Suhani? Done? Done. Acha, done. Okay, great. Okay, so now, okay. What should I do? Should I show you some videos or should I leave you early?
Miss. Anything's fine. Your choice.
My choice. Okay. Okay, let me show you 1 or 2 videos, then we'll wrap up. We'll wrap up by 6. Okay, do that. One minute, let me see which video was nice. I'll have to search. Okay, I can show you this one. This will be a recap. No, it's been a long time since we did hypertonic, hypotonic, etc. So we'll see that one. And then
How you take notes from YouTube like
this is a nice one. Now I
are
remember.
more likely to relocate. Today,
Right.
trapped in the middle of the ocean. You don't have any emergency food or water, but you also know that humans can only survive about 3 days without fluids. Should you drink seawater or not? Stick with us. We're going to find out and also review hypertonic, hypotonic, and isotonic solutions along the way. Cells have a very finite range of conditions in which they can survive. Too salty, too acidic, or too basic, they can't function properly. We're going to explore what happens when you place a cell in a solution that is much more salty, hypertonic, much more watery, hypotonic, or has an equal salt concentration, isotonic. Remember that cell membrane through, but not others. Usually, the cell membrane is fitted with special proteins through without expending energy. However, the ease with which the water molecules can cross concentration is a measure of how much solute there is per volume of solvent. In other words, whether the liquid is more salty or more watery. We'd usually expect that the molecules would follow a process called diffusion. They would flow from areas of higher to lower concentration. Eventually, they will reach equilibrium, where there will be an equal concentration on both sides of the membrane. However, in the case of a liquid like salt water, the salt molecule, the only particle that can move is water. Let's look a little more closely. Water molecules are highly attracted to salt molecules. They cluster around salt and really don't want to move. The positive parts of the water molecule stick to the negative parts of the sodium chloride, and vice versa. The interactions between oxygen and sodium and between hydrogen and chlorine are called ion-dipole interactions. Not stuck to a molecule of salt are far more likely to relocate. Today we're going to immerse a cell in 3 kinds of liquids: a very solute-rich liquid, a very watery liquid, and a liquid that's in between. Then we'll see what happens to the water molecules. In this first scenario, the liquid surrounding the cell has many molecules of solute in it. This liquid is hypertonic compared to the cell. It has a much higher concentration of solute particles and a much lower concentration of water. Unfortunately, the solute molecules can't pass through the membrane to reach equilibrium, but water molecules can. The molecules of water on the outside of the cell are going to be obstructed from passing through by the many molecules of solvent. However, the molecules on the in rushing out of the cell. Some water will come in, but the net movement, the overall movement, of the water molecules will be outwards and the cell will shrink. We know how pickles are made. Now let's immerse a cell in a much less concentrated solution, a hypotonic solution. Compared to the cell, it's much less salty. Some water molecules will exit the cell, but even more will rush in because they aren't obstructed by solute particles. The cell grows in size, and it may even undergo cytolysis and burst. Placing a cell in an isotonic solution, where the concentrations inside and outside are equal, is much more pleasant. Equal amounts of water molecules continue to pass in and out of the cell. The system is in dynamic equilibrium. Particles are continuing to move, but the net movement is 0. The cell neither grows nor shrinks and is much more likely. Seawater is incredibly salty, much more salty than our body's cells. Since it's such a hypertonic solution, if you immersed your cells in it, they would shrivel up and die. The real danger, however, lies in how your kidneys would react to the seawater. Your kidneys pulled from your cells.
Right, that is not really required. So this video, whatever we did, no, for osmosis and the different concentration, etc. type, just stick to that. Don't go to learn what is cytolysis, what is ion dipole, etc. That eventually you will learn. I mean, either this year or next year, you will definitely come across all of those terms also. But for now, the important thing is just to understand which direction the flow happens, and now you'll also know that why salt molecules are not moving, right? Inside to outside, or why they are not moving, only water is moving. All those things you can refer to from this video. So this, obviously I can't find a cut to cut video which has just the amount of things which you need to know, but yeah, there will be some extra things, so you can skip all that. Whatever we've done till there or that much, you make sure that you are thorough with. Okay. Okay, is there any other video? Let me see. Cell tour, I think, is a big one. I think it is. Okay. Okay. The others are all pretty big, so I will let you only watch it instead of me play. Okay. Right. Did you all go through the chapter once? Like, did you get a chance to revise or anything yet, or no?
In school, we haven't really finished with this chapter, so
Acha.
in fact, we are just at osmosis of the
Oh, you still at that.
Miss,
Okay.
we were at plasma membrane and nucleus stuff.
Oh, acha. Same.
Going
Almost
pretty
same.
slow.
Hmm.
We have a class tomorrow because there's just two classes for biology, physics, and chemistry in a week. So actually, maybe the portion. That would probably say.
Oh, okay. Take care. So you all also must be having summer vacations, right? From next week onwards. Or...
Yeah, from Monday.
Yes. Miss, tomorrow is the last working day.
Acha, great. ठीक है. ठीक है. Then I'll share this PPT today itself, and I'll share it like a PPT only, so you can see the YouTube links, etc. Also. Then you can go through the chapter at your own pace and pick up and learn whatever concepts you have to remember, etc. Right. Okay. Let me also give you summer vacation early then. Yeah. Okay. If you don't have anything, like no questions or anything, then we'll end the session today. Don't have much to do. Nothing.
Mm.
Okay. Have a nice vacation. See you all after the
Thank you,
vacation.
Miss. Bye.
Thank you, miss.
Bye.
Bye, miss.